about bacteria for a second. We see them as these really simple life forms, right? Their main trick is just splitting in two, making perfect, identical clones of themselves. And yet, they are absolute masters of evolution. They can adapt to almost anything we throw at them, including our most powerful antibiotics, and they do it at a speed that is frankly terrifying. So how? How do they pull that off without the genetic mixing that you get with sexual reproduction? Well, it turns out they have a secret, a kind of social network for trading genetic superpowers. Today, we're diving deep into the world of bacterial conjugation. Now, when you see an image like this, you might think you're looking at some kind of fight, but it's the exact opposite. What you're actually seeing is a bridge. This incredible micrograph shows two E. coli cells physically connected by this super thin protein tube. That tube is called a sex pilus. And right now, one of those bacteria is in the middle of injecting a piece of its own DNA directly into the other one. This process is called conjugation. It's the closest thing bacteria have to sex, and believe me, it is one of the most powerful engines for evolution on this entire planet. And that brings us to the central mystery, doesn't it? If bacteria are just making carbon copies of themselves, generation after generation, where does all the variation come from? How do they generate the incredible genetic diversity they need to adapt and evolve so ridiculously fast? This was a huge puzzle for early geneticists, and the journey to solve it uncovered this entire hidden world of microbial communication that we're about to explore. So, the answer to this paradox lies in a really cool concept called horizontal gene transfer. Now, normally we think of genes being passed down vertically, from parent to child. Horizontal gene transfer is, well, it's sideways. It's the movement of genetic material between two living organisms. Essentially, bacteria can swap useful genes like they're trading cards. And the most direct, the most intimate way they do this is through conjugation. To really understand how scientists first figured this out, we need to get a couple of key terms down. Okay, so auxotrophy. Sounds complicated, but the idea is simple. Imagine you couldn't make your own vitamin C. You have to get it from oranges or supplements to survive. That's basically auxotrophy. An auxotrophic bacterium is a mutant. It's lost the ability to make some essential nutrient for itself, so it can only grow if we provide that nutrient in its food. On the flip side, you have a prototroph. That's the wild type, the self-sufficient one. It can build everything it needs to live from just the most basic ingredients. This simple difference, who's a picky eater and who isn't, was the key that unlocked this entire mystery. All right, let's jump in a time machine. The year is 1946. The scientific world is in the middle of this huge debate about whether DNA is even the stuff of genes. And into the scene walk two scientists, Joshua Lederberg and Edward Tatum. They designed this incredibly elegant experiment to ask a very simple but profound question. Can bacteria even exchange genetic traits at all? The design of this experiment is just beautiful in its simplicity. Let's walk through it. First, they took two different mutant strains of bacteria. Let's say strain A couldn't make nutrients X and Y, but it was fine making Z. And strain B was the total opposite. It couldn't make Z, but it could make X and Y just fine. Now, the crucial part is, this meant that neither of these strains could survive on what's called a minimal medium, a petri dish with just the bare essentials lacking X, Y, and Z. For step 2, they just mixed the two strings together in a nice rich broth and let them hang out for a while. Then they took that mixture and spread it on to that bare bones minimal medium. Now logically, if bacteria only ever make clones of themselves, nothing should have grown. Period. But something did grow. Against all logic at the time, a few tiny colonies started to appear. And these weren't the mutant strains. These were prototrophs. Cells that could now, miraculously, make all the nutrients they needed all by themselves. This was the definitive aha moment. The only possible way these cells could exist is if strain A had somehow given strain B the genetic instructions it was missing, or the other way around. They had to be trading genetic information. The conclusion was just undeniable, and it was absolutely revolutionary. Bacteria were engaging in some form of genetic recombination. They weren't just a collection of isolated clones. They were a dynamic community, sharing and swapping genes. This discovery was so huge, it won Lederberg a Nobel Prize, and it immediately opened up a brand new, burning question. How in the world were they doing it? So the next piece of this detective story comes from Bernard Davis in 1950. Davis had a hunch. He suspected that for this gene swapping to work, the bacteria needed to be in direct physical contact. I mean, was one strain just leaking its DNA into the soup for the other to pick up, or was something more intimate going on? He came up with a truly ingenious experiment to find out for sure. What Davis did was he took this U-shaped glass tube, and right at the bottom he placed a very, very fine filter. Now this filter was the key to the whole experiment. It had pores that were big enough to let the liquid medium and all the dissolved chemicals and nutrients flow freely between the two arms of the tube, but the pores were way too small for a bacterium to squeeze through. So he put one of his oxotrophic strains on one side and the other strain on the other. They were sharing the same chemical environment, but they could never, ever physically meet. And the result? It was crystal clear. After letting them incubate, he took samples from both sides and plated them on that minimal media, and absolutely nothing grew. Not one single prototrophic colony. The verdict was in, and it was unambiguous. For this genetic exchange to happen, the bacteria had to physically touch. This wasn't some passive sharing. It was an active, contact-dependent process. Okay, so if they have to touch, what's the machinery making it happen? What's driving this cellular handshake? Well, further investigations soon zeroed in on a very special piece of DNA. It's called the fertility factor, or just the F factor for short. And this is where the story gets really cool, because the F factor is basically a mobile, shareable instruction manual for how to initiate bacterial sex. So the F factor is a plasmid, which is just a small, circular piece of DNA that lives inside the bacterium, but separate from the main chromosome. It kind of does its own thing. But it's also a special kind of plasmid called an episome, and that's its superpower. It has the ability to actually splice itself into the main chromosome. This little plasmid contains all the necessary genes, called genes for transfer, that act as the blueprints for building that sex pilus and all the other machinery needed to copy and transfer DNA from one cell to another. So let's see this in action, in its most basic form. You've got a donor cell, which we call F+ because it has the F factor, and you've got a recipient cell, called F- because it lacks it. The F+ cell extends its pilus, basically a grappling hook, latches onto the F- cell and physically reels it in close. Then, using this neat process called rolling circle replication, it nicks one strand of its F factor plasmid and starts to unspool it, feeding that single strand across the bridge. As that single strand enters the recipient's own enzymes get to work building the matching complementary strand. And just like that, the F- cell becomes a brand new F+ cell, fully equipped to pass the gift on to someone else. But what happens when that F factor decides to do something a little more dramatic? Sometimes, instead of just floating around as a separate plasmid, it uses its episome's superpower and integrates itself directly into the main bacterial chromosome. When that happens, it creates a very special type of donor cell called an HFR cell. That stands for high-frequency recombination. And as you can see here, an HFR donor plays by a completely different set of rules. When an HFR cell decides to conjugate, it doesn't just offer up a copy of its little F plasmid. Oh no, it tries to transfer its entire genome. This is conjugation cranked up to a whole new level. It's a high-stakes transfer with massive implications for how bacteria evolve. So inside this HFR cell, that integrated F factor still acts as the engine. It gets necked and it starts spooling a copy of itself into the recipient cell, just like before. But here's the thing. Because it's now physically attached to the much, much larger chromosome, it starts dragging the host's chromosomal genes along for the ride. The F factor is the engine, and the chromosome is this huge freight train it's trying to pull. Now, the connection between the two cells is pretty fragile. It takes about 100 minutes to transfer the whole chromosome, and that delicate little mating bridge almost always breaks long before the job is done. So what's the result? The recipient gets a big chunk of new chromosomal DNA, but it rarely gets the full F factor because that's the last part of the train to get transferred, and that means it stays F-. And this little flaw in the system led to an experiment that was pure genius. Scientists realized they could actually exploit this time-dependent transfer to map the bacterial chromosome. What they did was they'd mix their HFR and F- cells together, and then at different time points, say 8 minutes, 10 minutes, 16 minutes, they'd throw the whole mixture into a blender. Yeah, a regular kitchen blender. The violent mixing would just rip all those conjugation bridges apart, stopping the transfer cold. By then checking which genes had successfully made it into the recipient cells at each time point, they could figure out the precise linear order of the genes on the chromosome for the very first time. It's brilliant. Now, there's one final twist in this story. Remember how the F factor can pop in and out of the chromosome? Well, sometimes when it pops out, the process is a little sloppy. It makes an imperfect getaway, and as it loops out, it accidentally snags a piece of the adjacent chromosomal DNA and takes it along for the ride. This creates a new hybrid plasmid called an F' plasmid. It's the F factor, but now it's carrying some of the host's own genes as precious cargo. So when this new F' cell finds an F- recipient and conjugates, it transfers the whole package, the F factor and those captured chromosomal genes. Now, think about what this means for the recipient cell. It now has its own copy of those genes on its chromosome and a second copy that just arrived on the new plasmid. This creates a partially diploid cell, and the technical term for that is a merozygote. This process is an incredibly efficient way to rapidly spread very specific traits, like, for instance, a gene for antibiotic resistance, through an entire population. And that brings us to the big picture. Why all this molecular detail matters so profoundly in our world right now. Because this ancient system of genetic sharing, perfected over billions of years, has now become one of the single greatest threats to modern medicine. So let's just boil it down. You can think of conjugation as the roaring engine of bacterial evolution. You see, mutation creates new genetic traits slowly and randomly, but conjugation is the superhighway for sharing the winning traits almost instantly. It's how bacteria trade the blueprints for surviving our best antibiotics. A single bacterium can evolve resistance on its own, and then, through conjugation, it can share that superpower with its entire community, and even with completely different species of bacteria, creating dangerous superbugs practically overnight. I really love this quote because it puts everything into perspective. This ancient genetic network is faster than evolution. What does that really mean? Well, traditional evolution works vertically, from parent to child over generations. It's a slow, steady climb. Conjugation works horizontally. It's like sending out a software update to the entire existing population all at once. It's a global, invisible, and unbelievably rapid communication network operating on a scale that's just hard to wrap your head around. And so that leaves us with a really critical question for our future. We've cracked the code. We understand the very mechanism by which bacteria arm themselves against our best defenses. But knowing how the network operates is one thing. Being prepared for the next evolutionary superpower they decide to share across it, well, that's another challenge entirely.